Hi, thanks for coming to my poster. My name is Nygard. I'm a PhD student at the University of Ottawa in the lab of Matthew Lovely Adam, and I'm presenting today my poster entitled Graph Model of Chromosome Conformation Capture Data Allows for Statistical Assessment of Gene Functional Annotation Clustering in Folded Nuclear Chromatin. So to introduce the topic, we can have a look at this top left panel here, where we talk a little bit about how chromatin in eukaryotes, particularly we're looking at human cells, is known to fold tightly and non-randomly into a hierarchy of nested structures. This ranges from the open and closed chromatin compartments that house active and inactive chromatin respectively, down to the chromosome territories that segregate individual chromosomes into discrete domains, and newer structures that we found that are even finer grained, such as laminar associated domains and topologically associated domains, which we're really just learning about in the last decade and seem to have an essential role in transporting genes from the active and inactive states. But as for the organization and the overall content of these TADs, there's still a lot to be discovered. So the technology that was used to find TADs and is frequently used to explore this type of data is called conformation capture, or 3C. Particularly one of the most popular methods these past several years has been Hi-C, which is a high throughput version of this technique. And basically the way that it works is you just fix the nucleus in place using formaldehyde, and you chop it up into roughly even restriction fragments using a general acting restriction enzyme, such that you can religate the pairs with a biotin tag, so that you get these chimeric reads that can then be read by a next generation sequencer. And this basically gives you a list of contact frequencies so that you know which loci within the genome were in frequent contact with one another. And the idea being that if two loci are frequently contacting, then you can basically infer a probabilistic distance between any two loci and thereby get a 3D sort of spatial model of the nucleus as it existed at the time of the Hi-C experiment. So as I mentioned, this technique has been used plentifully to uncover a number of hierarchical structures within eukaryotic genomes, particularly human genomes are organized in this way. And we've learned increasingly fine-grained information about the structures themselves. But as for the relationship between gene function and storage within these structures, we're really only starting to brush the surface of this. So the goal of this project is to create a computational tool that allows for that type of analysis. And in particular, the software that we developed for this project, called Gothic, takes Hi-C data, converts it into graph structures or network structures, such that we can utilize the breadth of methods available in graph theory in order to assess clustering and assess these structure-function relationships. Furthermore, in this project, we want to use this tool to assess the clustering of gene ontology terms as our functional set of interest, and apply those to Hi-C datasets that are publicly available on ENCODE. So just to give an overview of what our workflow sort of looks like for this project, we basically take the Hi-C contact information from ENCODE or whatever experiment, we turn it into this spatial chromatin network, basically a skeleton of the network that is unannotated but represents the 3D structure of chromatin within the nucleus. We annotate that network with a functional set of interest. As I mentioned, this is gene ontology terms for us. And once we have the annotated network with edge weights corresponding to those contact frequencies, as I mentioned from the Hi-C experiment, we can then assess the clustering of functional terms within the network. Basically, this gives us the power to assess whether genes that are related by some functional term, maybe some metabolic pathway or some other shared function of interest that might, be representative of some sort of disease state or a study of interest. And basically by looking at those clustering patterns, how they tend to segregate and cluster within the network can give really meaningful insight into how 3D topology affects overall gene transcriptional control. So we utilize 3 clustering methods to apply to this network, and it's important that we capture a breadth of clustering methods just because these methods all sort of capture different facets of the data. For example, we use weighted total pairwise distance, which is able to take a query set of nodes within the network, so a number of loci, say all loci that are annotated with a given gene ontology term, and by comparing the sum of the shortest pairwise paths of that set to a null distribution of a set of the same size, we can determine whether a set of nodes is statistically significantly clustering within 3D space in the nucleus. And by comparing this to alternative approaches like the Markov clustering algorithm and spectral clustering, which instead scan for the natural clusters in the network using comparable but different methods, and then looking at those sort of quote-unquote natural clusters in the context of gene enrichment and looking for those functional enrichment of gene ontology terms within those clusters, we can sort of get a comparative set of functions that are clustered within the 3D nucleus at the time of the Hi-C experiment. And results-wise, our approach has identified a number of interesting things and have sort of validated the reason of going forward with this type of approach. So using the weighted total pairwise distance method, we found that consistently in a real biological Hi-C network terms are more consistently significantly clustered than in a random null model network where functional terms are segregated randomly. So this basically gives us the idea that some effect is driving functions to cluster within the nucleus. Loci tend to cluster if they share a functional term. And we also show that this is not only due to linear proximity along the genome. So this is good supportive evidence. This sort of gives us an idea that our software is working. And with this, by comparing all these three clustering measures, we got a large list of terms for this MEC data set that we're working on that do cluster and are significantly enriched within the nucleus. By looking at this overlap set that showed up in all three of the clustering algorithms, we found a small set of Go terms that seem to be picked up regardless of what type of clustering approach we're using. Namely, these include protein DNA complex assembly and nucleosome organization, which are two terms that actually have a lot to do with gene organization and chromatin rearrangement on their own. So it makes sense that these terms are clustered because they would have to be sort of more central within the nucleus. And this is validated by literature that these terms are associated with chromosomal rearrangement and so should be active in the active compartment of the nucleus. So it makes sense that they end up being clustered. So these are all positive results. And now what's left to do with the project is to take a number of comparisons, basically looking at different types of cell lines, say a healthy colon tissue versus colon cancer tissue, male versus female cells, cells of different types, comparing breast tissue to colon tissue, etc., and probing for overarching patterns and terms that cluster specifically to cell condition and those that seem to sort of universally cluster within the cell. And so we're excited to present the software and present the preliminary results that we have gotten from this work. If you have any questions, feel free to add them on my page here on ISMB or come talk to me at my poster. Thank you.